First off, thank you guys for coming, and we're really pleased to have you here. And over the next few minutes, I'm going to tell you guys some really basic things about neurodevelopment, degeneration, and disease, and how stem cells can help to broaden our knowledge of these processes so that we may expedite identification of translational applications. So, one of the first things that you have to do when you're trying to establish a disease model in the dish is to assess the complexity of the disease and ask how that complexity may affect your ability to study that in the culture dish. So there are some relatively simplistic diseases or low-hanging fruit, such as diseases where we have a monocellular deficiency, such as Parkinson's disease. You all very well know that in Parkinson's disease, a specific type of neuron in the substantia nigra degenerates, and you have a lack of dopamine production. This is an attractive target for stem cell-based therapies and disease models because we only have to generate a single cell type and study that cell type. Another type of low-hanging fruit is those of monogenic diseases. So in these cases, such as with Fragile X, it's a single gene that is defective and causal of the disease. So in that case, we only have to try to study a single gene and how to correct the aberrations that come from the mutations in that gene. Now, some relatively higher hanging fruits on the stem cell-based platform would be those of multicellular deficiencies, such as Alzheimer's disease, where eventually almost every cell type in the brain becomes affected, and also multigenic or idiopathic diseases, where we don't really, there's a number of genes that are implicated in the causality of the disease, but we don't really understand how it's happening. An example of this would be schizophrenia or most cases of autism. Now, Alzheimer's disease is incredibly prevalent in the United States. About 5.4 million people are affected, and that's 1 in every 8 people over the age of 65. And it's also, you guys know, it's a kind of a painstaking disease that takes some time. So for the pathogenesis of Alzheimer's, starting here with a healthy brain, we first have identification of these two hallmark characteristics of the disease known as amyloid plaques or neurofibrillary tangles. These plaques are just protein aggregates that can be detrimental to the way that the brain flows, and also these neurofibrillary tangles are just the cellular structure kind of gone awry. And these are the two hallmark characteristics of Alzheimer's disease, and actually a number of other degenerative diseases in the brain as well. So the progression of the disease is slow and expanding over time, starting in a rather localized area in the temporal lobe, but over time becomes more expansive throughout the temporal lobe, beginning to affect the back of the brain and the parietal, and kind of the command center of the brain, the frontal lobe, and eventually, at the time of death, affects most of the brain. As you can see, this is a degenerate Alzheimer's brain, which has lost a lot of its biomass. And if you were to project the normal brain behind the Alzheimer's brain, you can see that it has reduced in size over the progression of the disease. Now, my interest in Alzheimer's comes from personal experience in my family. And I wanted to just highlight a couple of structure-function things with the brain so that you can try to understand some of the behaviors that you see as the disease progresses. So there's 4 areas of the brain or 3 areas that I'm going to highlight. The first is the temporal lobe, which is, amongst other things, responsible for emotion, memory, and speech. In the back, we have a parietal lobe, which is cognition, orientation, and recognition. And again, the frontal lobe, as I said, is kind of the command center of the brain, where we have reasoning and problem solving. So to go back through this time course of the disease, with my grandfather, I can clearly remember in the early stages just kind of some short-term memory loss and things such as going to the drive-through, paying for your food, and then forgetting to get the food and driving off. And we would laugh it off at the time and think that he was just silly, but goofy. It was a early signs of a larger problem.
I saw some overworked scientists do that.
Yeah. This is actually not the disease, this is just my grandfather. But this is later on, when the disease was likely spreading throughout his brain and his behaviors were becoming more erratic and problematic. At this point, we had some cognitive problems, as well as orientation. He was getting lost in places that should have been familiar, and also recognition. One particular instance that still to this day fascinates me, and the way that the brain works and degenerates, is I can remember walking into the bathroom. My grandfather was trying to brush his hair with a toothbrush and asked me why it wasn't working, and I said, well, Papa, you know, it's a toothbrush, it's not for your hair. And immediately he got it and was just, you know, so embarrassed and made me promise I would never tell anybody. So I'm telling all you guys.
Here you are. That's
and,
nice.
You know, it's kind of one of the most devastating parts of this disease is that, to some extent, the affected patients have to witness their own demise. And by the time he died, this is just a couple of months before, you can see that he's almost entirely stoic. His eyes were closed, and it rarely said anything, and when he did, it didn't make much sense. So a lot of progress has been made since the time of my grandfather's death. And one of the most impressive studies, I don't know if any of you guys have ever heard of the Nun Study. Anybody heard of this study? Yeah. So this is really just an amazing, amazing study. In the study, there was nearly 700 nuns from a single place, the School Sisters of Notre Dame. And David Snowden at the University of Minnesota and later at the University of Kentucky was doing an epidemiological study on these nuns and followed them for many, many years. This ended up being arguably the most controlled epidemiological study ever conducted, and it's still ongoing. The reason why it was so controlled is that all of these women lived together, ate the same food, lived the same lifestyle, never drank, never smoked, had autobiographical information from the time they were 18 on, and David was able to follow them throughout their entire lives. More fantastically, more than 90% of the women donated their brains for research. So if you have nearly 700 women, 1 in 8 contract the disease, you have a number of brains to compare to each other. And one of the most striking findings that came out of this was these characteristic plaques and tangles, which were the focus of much of Alzheimer's research for years, turned out to have very little correlation with the or predictive power in the extent of dementia and Alzheimer's disease. In fact, some of the most plaqued and tangled brains were from nuns that had no signs of dementia or very little signs of dementia. So this really changed the way that we thought about Alzheimer's disease and what these plaques and tangles were. So for years and years and years, we were all focused on this. But with this study, it became evident that this is not where the disease was happening. They're a quarantine of disease that has come and gone.